Today is Monday, June 9th, right? Unbelievable that we're already toward the middle of June. And we have an exam on Wednesday. That exam is going to cover what? 3, 4, and 5. Chapter 3 was what? All about cells and transcription and translation and replication. We mentioned some of those special processes that cells do. For that, you want to make sure you know the organelles, you can recognize them, you know what their functions are. Then chapter 4 is histology. And we've been talking about that in lab. You've all been quizzed on it, or will be by this evening. You've all been quizzed on histology. What we focused on in in lab has been 6 different epithelial and 6 different connective tissues. That's about as far as I got also in lecture. I'm going to add to your repertoire just a little bit today as I show you a couple of images of muscle and of nervous. That will finish up chapter 4 for us. And then chapter 5 is over skin. And I'll be discussing the structures of skin with you today. Those 3 chapters, 3, 4, and 5, will make up the exam on Wednesday. That exam will be a full-length exam. That's all we'll do while we're here together. I just finished it, almost finished it. It's not much longer than the last exam. You're not going to need all 2 hours, but just know that we'll start it at 4. I suspect you'll all be done by 5:30. You'll be done a little bit early on Wednesday. And then after Wednesday, your major focus will be on your lab exam next week, won't it? So it's a really, really busy 10 days between now and then. You've got a lecture exam on Wednesday. Next Wednesday, a week later, you have a lab exam over the first 5 labs. And there's quite a bit of material there that is just now coming into focus for you with the bones, the bone markings, and next week the muscles. So I get it. There's a lot going on. So hang in there. You'll be fine. It's going to take a lot of diligence, though, between now and next week to really let this material sink in.
When did you say the lab exam was?
Next Wednesday.
So in lecture? During lecture? So like those of us that have lab on Monday, it'll be on Monday?
No. No, the Monday students were told the first week you come to lab next Wednesday from either 2 to 4 or 6:30 to 8:30 after lecture. So next Wednesday is, for Monday lab students, next Wednesday is different for you as has been on the schedule. You will come to lab on Wednesday, either 2 to 4 or 6:30 to 8:30 in the evening to take your lab exam. Then at the end of the semester, Wednesday students will be coming in on Monday. So each group will get to be inconvenienced equally. Don't feel special for having to do something extra at this point. So we were discussing tissues. I also need to go ahead and pull up our last little bit of vocabulary, because don't forget we also have what vocabulary on this exam? 21 through 40. And so far, I believe I went through 35. Does that seem right with all of you? Did I get through glia before and gingivo? I believe that was where I got. So let me finish up the last little bit of vocabulary with you as well. There's probably going to be 15 terms or so of vocabulary on your exam on Wednesday. And 15. Just like before, about 10% of the exam will be from vocabulary. So let's take a look at this term glom. We'll see when we get to the kidney, a structure in the kidney called the glomerulus. The glomerulus is a ball of capillaries that's very important in the filtering of your blood. So for now, just know that glom, when you see that term, refers to ball. Glosso, tongue. Something given hypoglossal under the tongue. Gluten, glue. It's what kind of glues your bread products together, but also just means glue. Agglutination, when we talk about antibodies and antigens and blood typing a little bit in a few weeks, I'll mention the word agglutination, and that is how molecules stick together like glue. Glycolysis, glycolysis, the breakdown of sugar is a very important process in our getting energy for our metabolism. For now, just know that glyco means sugar or sweet. Nausea or gnosis, knowledge. If you're given prognosis, you have knowledge in advance, right? In advance of something. Gonads are your sex glands, the testes and the ovaries. And gland, we'll see that term today, refers to a grain or a particulate or a little particle-like structure, and we'll see a layer in the skin called the stratum granulosum. There are also blood cells called agranulocytes because they don't have granular-like or particulate-like features. Gram, a recording or a picture of something. So an electrocardiogram, a recording of your heart rhythms. Also, if you see a term endography, similar. Gravido, pregnancy. You might see a term like gravidocardiac referring to some cardiac event that occurred during pregnancy. And then gynecologist, right, a person who studies or specializes in the treatment of female issues, mostly reproductive issues. Halu, the great toe, the hallux is your great toe. If we're dealing with numbering our digits, that would be digit number 1. So your big toe and your thumb are always referred to as number 1. Your pinkies are number 5. Haplo and hapto, we've seen this term before in passing. Haploid or haploid means a single in meiosis. Remember we talked about how meiosis is a cell division in which we make our sex cells, the egg or the sperm. And in doing so, we end up with cells that only have one copy of all the chromosomes. They are said to be haploid, whereas the rest of the cells in your body that have 2 copies are said to be diploid. Hemato or hemo, both of those terms refer to blood. What process is occurring in your bones that we talked about in lab this week, in last Wednesday? What process is happening in your bones? The formation of making your blood cells? Hematopoiesis or hemopoiesis. So we'll start talking more about that as we get to the blood. And then hemi means half. What other term have we seen that means half? Didn't demi come across our list? All right, so demi and hemi and semi, right, we'll see later on, all mean half. Hemiplegia is paralysis of half of the body. Lastly, for this exam, hepato, the liver, hepatitis, an inflammation of the liver, maybe caused by some disease of the liver, inflammation. Hetero, different. Histo, we're talking about histology, right? We're studying tissues. So histology, the study of tissues, not to be confused with histero, which is referring to the uterus or the womb. Now, look at the spelling here. Depending upon if you're spelling it more British or more American English, you'll see hysterectomy or hystero with a Y or an I. They're both proper in different sides of the pond. The way I remember this term, and guys, you'll appreciate this, only women can be hysterical. Did I say that out loud? Okay, it works though, doesn't it, right? So hystero referring to the uterus or the womb. Of course, that's just in jest, right? Now, so that finishes our vocabulary, 21 through 40, right? Vocabulary on exam number 2. That also means we're 40% of the way through the course because there are 100 slides. So as we move through these slides, man, this is like one of those little thermometers that, builds up as we go through the course. So we're now 40% of the way through the material. Hard to believe, but that is where we find ourselves already. Anyone concerned about vocabulary? You know what to expect this time. Oftentimes I'll put together 2 of these terms, maybe even 3 of them in a sequence. And what would you have to do? Tell you what each one means in the order in which I give them, right? So you'll break the word down into the 2 or 3 portions and fill in the blank. This is definitely fill in the blank kind of information on exam number 2. Let me just begin to move through these slides quickly as a reminder. We're trying to get an idea of the tissues, and we discussed how in early development, egg and sperm coming together to form the zygote. The zygote then undergoes cleavage or division, and we get multiple cells. Those cells must then travel down and adhere to the wall of the uterus to set up pregnancy for the next 9 months. That ball of cells will begin to hollow out. It'll begin to envelop and become a 3-dimensional structure, and that 3-dimensional structure is called the gastrula, and that gastrula sets up 3 layers of cells, doesn't it? And what were those 3 layers? The inside layer, shown here in yellow, is the endoderm. The blue on the outside is the ectoderm. We know ecto means on the outside. And in the middle was the mesoderm and the mesoderm. And from each of those layers, different tissues become evident. So what did we say was formed from the ectoderm? The outer layer of that gastrula, of that blastula, will become the nervous system and will become the epidermis, the outer layer of your skin. That kind of makes sense, the outer layer. What about the inside layer? The endoderm will become your tubes, right? That'll become your GI tract, your digestive system. It will become your respiratory tract and your windpipe. That kind of makes sense, too, as we think about our body having these tubes within us. And then lastly, the middle, the mesoderm, will become the muscle and all of the connective tissues. Now, this is it's a close enough for what we're doing, right? So that's a basic way of thinking about it. Your mesoderm, the middle layer, what's shown in this picture in orange, will become eventually your muscle and connective tissues. Now, what word do we use to describe cells becoming different from each other and becoming more specialized? Differentiation. Right? So tissues become more differentiated. They undergo differentiation so that certain cells become skin cells. Certain cells become kidney, some liver, some brain. And there are over 230, 250 different kinds of cells in your body. They all become different. And then we discussed also, though, last lecture about tumor cells. What do tumor cells do? Typically, they de-differentiate. They become more what? Simplistic, more adolescent, more childlike. And children grow faster than adults, and so too these de-differentiated cells grow more out of control. Right? And they grow and they lose their contact inhibition and they tend to metastasize and some of those things we described when talking about uncontrolled cellular growth. So that's a quick reminder about the layers. And then basically we were doing a quick march, weren't we, through the tissues. And I spent the majority of the time on the epithelial tissues and on the connective tissues because that's really where our focus is right now. We'll deal with muscle tissue in much greater detail when we get to muscle later in the course, exam 3. And then when we get to nervous system, exam 4, we'll deal with nervous tissue. So we're setting up this hierarchy, if you will. And a reminder that epithelial tissues are the only type of tissue that comes from all 3 of these germ layers. So you will find the epidermis, right, the outer layer of your skin, comes from which layer? This makes sense, guys. So the outer layer of your skin, the epidermis, comes from which of the germ layers? The ectoderm. And the lining of your gut tube, the lining of your pharynx, of your throat and of your windpipe, your trachea, and the lining of your, well, we'll go with those structures. That would come from what layer? The endoderm. And then when we think about the peritoneum, the pleura, and the pericardium, those are epithelial layers that are actually derived from the mesoderm. So the epithelial layers of your body are actually derived from all 3 of those germ layers, whereas all of your connective tissues are mesodermic. All of your muscle is mesodermic, and all of your nervous tissue comes from the outer layer, the ectoderm. We spoke mostly of characteristics we already knew about in tissues. The fact that epithelial tissues are normally avascular, they're highly mitotic, they are a selectively permeable membrane. We discussed also in this picture what are the connectors between these cells. Remember, epithelial cells are tightly packed, and there's very little space in between them, but the cells do communicate one with another, and they're held together tightly to create these barriers. So there are tight junctions, right? And you can ignore the Latin names on this particular slide, but there are the tight junctions. They were more apical, more toward the outside. There were the adhering junctions that were deeper down. Kind of looks to me like Charmin, right? If you look at layers of toilet paper and the kind of how they're held together. And then there were the desmosomes that were like little spot welds or rivets for extra stress areas. and then there were the gap junctions, which allowed for communication of molecules from one cell directly into the next cell. The other thing I like about this particular slide is that it shows both layers of the basement membrane. Remember we discussed both layers? The layer made by the epithelial tissues was called what? The basal lamina. And the layer below it, still part of the basement membrane, but made by the connective tissues, is the reticular lamina. So 2 layers. Most books just show us 1 layer. And the reason they only show us 1 layer is that when you're using a light microscope, you can only see 1 layer. You cannot see both of these layers in a light microscope. So it's kind of one of those academic issues right now. But in reality, that basement membrane is partially made by the epithelial tissues above and partially made by the connective tissues below. We then went through different examples of the epithelial tissues. I showed you examples of each. Make sure you have looked at these pictures. I'm very likely to use some of these exact images when asking you to identify these tissues, so do be familiar with these. And then we spent a little bit more time talking about glands, because remember that glands themselves are largely epithelial. And we saw that glands release products by 3 different mechanisms. And what were they? Remember merocrine? And how are you going to remember merocrine? Crin means to secrete, remember? And marrow or Mary, I said what? The cells are remaining alive or happy, they're merry. And so they're releasing their products without killing themselves. And merocrine secretion is what we see in tears and in sweat and in your digestive gut juices. And then there was holocrine. In holocrine secretion, what happens? The whole cell disintegrates or is released. These secretions are very thick and oily and lots of particulates in it, which lead to clogging up pores. And this is the type of secretion that is characterized by your sebaceous glands, your oily glands of your face. It makes sense why they can so easily clog up the pores. And lastly, there was apocrine, which was really a combination, isn't it? Just the apical portion of the acinar cells is pinched off and is released, and breast milk is released in this methodology. So breast milk is not just water, there's some particulates in there mixed in with the cellular products. Then we moved into connective tissues, and we went through the connective tissues that we already know and love and adore and can identify. We are reminded that all connective tissues, it didn't matter if it's tendon, ligament, fat, blood, cartilage, or bone, all belong to the same family because they all are composed of what? Loosely packed cells, surrounded by stuff called ground substance, and in that ground substance one finds proteins, collagen, elastin, or reticular fibers. We looked at our different tissues, and it brought us, as I recall, to a slide that said membranes. Do we all agree that's where we stopped last time? Okay. So when you hear the word, any questions, first of all, on that quick review of what we discussed last time? Anything at all? Quick whirlwind tour. Yes?
In lab, you said to know the 6 different parts.
In lab, I told you to know the 6 different structures of bone.
But on the slide, you had us cross out.
I did. That was the 7th. And in lab this week, you'll be asked to know that 7th. So what were the 6 structures to know from bone? You had to know that the entire cylindrical structure was called an osteon, number 1. Number 2, in the center of it, there was a central canal. The tissue was made up of rings called lamellae. Making up the rings were structures where the cells hung out. Those are the lacunae. The cell types in those lacunae are osteocytes, and those cells communicate through little cracks or canals called canaliculi. There's our 6 things you had to know. In addition, this week in lab, or last Wednesday, I introduced Volkmann canals. They'll be the 7th structure you'll need to know from bone, and they are the perforating or the perpendicular canals that connect one central canal to another. Okay, so I'll mention those again in lab. Volkmann's canals. That'll make your list of special things to know about bone up to 7 things. When you hear the word membrane in anatomy, I want you thinking something that separates, a divider. And there are 4 basic membranes in anatomy. Now, I know when I first hear the word membrane, I think, oh, a cell membrane. Right? A cell membrane, the phospholipid bilayer with the integral proteins, and that's what every cell has around it. But also when we're talking about membranes in an anatomical standpoint, we're talking about a sheet of epithelial tissue, usually very, very thin, but not always, with a layer of connective tissue down below it. And you've already heard of at least 3 of these different membranes. Number 1, mucous membranes. Describe to me your mucous membranes. Name for me a mucous membrane. The oral cavity, right? The inside of your mouth. Lining of your gut tube, right? The anus, the vagina. So what is it, the nasal passages, what's common there? They're all open to the outside world, right? They have a direct connection to the outside, to the inside world. And the way I always think about them, a mucous membrane is someplace where I can put Tabasco sauce and it's going to burn. I'll let you imagine. Okay? So any place where you are going to burn from hot sauce, that's going to be a mucous membrane inside your mouth, and the other places we're not going to experiment. But trust me. Now, what about serous membranes? We talked about the 3 different serous membranes in Lab 1, the 3 Ps, the pleura, the pericardium, and the peritoneum. We call them serous membranes. They are really just a thin layer of epithelium with a little bit of connective tissue down below. And you know that these membranes were made up of a double layer, right? Remember the parietal layer and the visceral layer. This is old stuff. Those layers are also referred to as the mesothelium. Right, so this is the epithelium made from that mesoderm, from that middle layer developmentally. The third one we'll be discussing today, cutaneous membrane. Fancy word for your skin. So this is your epidermis and your dermis. Epithelial sitting on top of connective, right? That's what your skin is. It's an epithelial layer, your epidermis, sitting on top of a connective layer called the dermis. Again, it's protective, it's a barrier. And we know what kind of epithelium is found here, don't we? The outer layer of your skin is characterized by having what? Keratinized, stratified squamous. Let me go back. No, that's fine. Keratinized, stratified squamous, that's fine. We'll keep thinking about that right now. The 4th membrane that I have not described, but you may have heard of, are your synovial membranes. These are membranes going around your joints, around your knee, shoulder. A lot of your freely moving joints have synovial membranes. This is, again, cells that are dividing and creating a sac-like structure that give your joints lubrication. So again, I bring this term up, body membranes, because it's the coming together of the epithelium and the connective tissue, isn't it? It's where we have 4 different examples of epithelia sitting on top of connective, creating what we call body membranes. Now, the last 2 types of tissues, this is going to go much more quickly. You're going to learn a lot more about this over the next weeks. But I do want you to have a visual picture of this burned onto your retina. Muscle tissue. What is it that makes muscle tissue different from nervous or epithelial or connective tissue? You tell me. You know this. Yeah, muscle tissue contracts. It changes shape. That's its claim to fame. That's what makes it unique in the body. Now, this muscle can change voluntarily or involuntarily, meaning you can have control over it. Your brain says move it, voluntary, or there are muscles in your body which do their job without your instructions. Thankfully, your heart, right? Your cardiac muscle is beating for you 24/7. You're not having to stay awake. So we know that the cardiac muscle would be considered involuntary. And regardless, we're moving things. We're moving the muscles of our skeleton. We're moving the products of digestion. We're emptying our bladder. Or we are pumping blood through the heart. Regardless, that is being done by muscle. Now, there are 3 types of muscle. A lot more about this next week in lab, or this Wednesday in lab, we'll describe these muscle types as well. Muscle types include skeletal, cardiac, and smooth. That's all I really want you to know, is that there's 3 different types of muscle. I'm going to show you an image of each one. I promise you that on Wednesday on the exam, I will show you these exact images. So right now, you just need to get these images ingrained in your brain to remember them. Again, skeletal muscle is voluntary. You choose to move the muscles of your body. This week and next week, we'll be learning about 25 or so different muscles in the body. And every muscle we're learning about in lab is a skeletal muscle. Your biceps brachii, your gastrocnemius, those muscles are all voluntary muscles. Cardiac muscle, the heart, we don't tell it to contract. It is involuntary. And also there is smooth muscle. This is muscle that's around many of your viscera. It's also called your visceral muscle for good reason. So it's around your internal organs. You have no control over it, but thankfully it's always doing its job, helping with digestion, helping with other processes. So let's take a look at these 3 images. I'm going to help you tell a story to remember these. When you look at this, I hope you'll agree with me, it doesn't look like any epithelial tissue we've seen. It doesn't look like any connective tissue we've seen. It's its own special tissue. This is what skeletal muscle looks like, and this is the picture I want you to know for now. What do you see? Again, when we saw adipose, we said corn pops or marshmallows. When you see this picture, does anything come to mind? Tire tracks. I love tire tracks. Does everyone see tire tracks? So let's tell a story with tire tracks. So if those are tire tracks, someone had to do what to make the tires move? Step on the gas. And to step on the gas, somebody had to make a voluntary choice to do that. So this is voluntary muscle. This is your skeletal muscle. Right? So you see the tire tracks, think, okay, I see those striping patterns. This is skeletal muscle. We'll deal with what those stripes are later. Okay, that's not my point now, but I do want you to recognize this as skeletal muscle. Keep that story in mind, that this looks like tire tracks. Number 2, the second kind of muscle I want you to know is cardiac muscle. What do you see in this picture? Anything come to mind? Something a little bit different here. Yeah, I still see those striae. I still see some striping patterns, but it's not as dramatic. But I do see these perpendicular lines. See these blue lines? Now, those lines are called intercalated discs. We'll deal with those later. But those intercalated discs give this tissue, in my mind, sort of a bamboo look. I see bamboo shoots. Do you see them? Kind of moving from down to upper. So it looks like some bamboo shoots are growing. Well, what animals eat bamboo? And we love pandas. So cardiac muscle, right? So we love the pandas, the heart, cardiac. Just pull that story together and you won't forget it. So we've got something that looks like bamboo in this picture. And again, this is the only picture I'll ask you to know for Wednesday. This is cardiac muscle. It is involuntary. We don't choose to make our heart do its job. And we'll deal with other characteristics in lecture and lab at a different time. Finally, smooth muscle. What do you see here?
S's.
Say it again.
S's.
S's? Worms. I love the worms. So if you had worms, where would they be? In your stomach. And what kind of muscle is lining your stomach? Smooth muscle. Okay, so when you see little parasitic worms, right? You've eaten some bad pork, right? So if you see little parasitic worms, you see little worms, right? For now, keep the story simple. Just use this little story to keep a grab of it. Little worms, parasitic worms, where would they be? They'd be in your stomach. Your stomach is surrounded by this smooth muscle. That's what I want you to know from muscle when it comes to our time together on Wednesday. Just recognize these 3 images. That's it. One slide on nervous tissue. If I were to ask you, what is it about nervous tissue that's different from muscle, connective, or epithelial tissues? You know that nervous tissue does what? It doesn't change shape. It doesn't cover layers. It doesn't connect us, store. Instead, what does it do? Say it again. Communicates. It's able to send electrical signals through our body very, very quickly. And we'll deal with the nervous system in greater detail. For now, I just want you to realize, yeah, the nervous tissue is sometimes called the neural tissue. It's made up of a bunch of cells called neurons. We'll break those down and know those cells really, really well here very soon. And it's specialized because nervous tissue can detect signals, what we would call stimuli. It can pick up light or sound or vibration, and then it can send those signals rapidly through the body through electrical impulses, and as a result, have our body respond to these stimuli. This tissue is specialized, or I shouldn't say specialized, but is localized largely in our spinal cord, in our brain, but it's everywhere. Right? We've got nerve endings in our fingers and all over our skin and all over our organs. So it truly is everywhere in our body. And some of these neurons are the longest cells in our body. I mean, incredibly long. You've got single cells that go all the way from your spinal cord down to your big toe. In a person my height, that's 1 meter long, right? You've got cells that are 3 feet or longer that are actually able to communicate. So really, really cool cells. We'll talk about them when we get there. So this also means that you have to get rid of the idea that cells are all little round balls. I know that the cells I had you learn were these little spheres with a purple nucleus, right? A green Golgi and an orange mitochondria. I know that. But you also have seen epithelial cells, and you know that sometimes they're columnar and cuboidal and squamous, and you're going to be seeing other cells as well. So I don't want you getting locked in that all cells look exactly the same. And the neuron is a very strange cell when compared to others. Here's the only image I want you to recognize when it comes to the nervous tissue on Wednesday. Doesn't look like anything else. I'm not even going to go into everything that this picture includes because that's really beyond the scope of today's time. But these are neurons, these larger cells that we're seeing in here, and these are the cells that are quickly communicating with different parts of the body. And that's really all I'm going to say about that. Make sure you recognize that particular image on Wednesday as the nervous tissue. That's the only image. So now, how many tissues do you need to know for Wednesday? 6 epithelials, the same from lab. 6 connectives, the same from lab. 3 muscle, right? I gave you those 3 pictures, the skeletal, the cardiac, and the smooth. And now a image of nervous tissue. So that's a total of 16 different pictures. And again, I will quiz you from these pictures in the PowerPoint. So when we get a little more sophisticated, I'll spread out and I'll show you different images. But for now, you're dealing with just these images on the screen. Now you tell me, what has made histology for some of you more difficult? What has made histology a little bit more difficult? Some of you are not big fans of this yet. What is it? Terms, okay. You've never perhaps done this before. You've got 16 different pictures that you've got to memorize. If this were a Western civilization or an art history class, right, you'd be, trying to figure out what that picture was and who's the author and what era is it from. And it's sort of the same idea. This is a very visual portion, and some of us aren't as good visual learners as others. The images I'm showing you are going to be very much the same, but as you're doing this in the lab, we shared some frustration because the tissues are cut randomly, and the sample we have in our microscope slide may not always look like our neighbor's sample, and we've got to do some hunting and a little bit of looking around to find these things. So it takes some repetition. And as you look at these tissues over and over, you'll begin to see the connections. But if you look at them just once or twice, you may not notice those connections, and you'll be more randomly guessing when it comes to the exam. Remember, you're going to see this on Wednesday for the lecture exam. You're also going to see this, some of you already saw it in your lab quiz, and you're going to see it again on your lab practical exam. And I promise you, it doesn't go away. You'll be hearing about and identifying histology and histological images all the way through the rest of your anatomy career. So this is foundational. Just like knowing the bones, you need to know your tissues. So that's where we're heading. So I'm just trying to encourage you, but I recognize that as you first look at these things, it gets tough. Again, you need to be able to recognize them, know their general location in the body, and any particular changes that were, or any particular structures or special features that were described. While we're talking about tissues, let me give you 5 or 6 terms that describe changes in tissues. The first one, metaplasia. Metaplasia. This is an example of epithelial tissues that change. A great example of metaplasia is in a smoker's trachea. At first, I mean, why do we have, think about it, what kind of epithelium lines our trachea normally? Pseudostratified columnar, and on those cells are a lot of cilia, those goblet cells for the mucus production. And as a person begins smoking, they cough, and their body basically gets pissed off after a while and says, screw this, and stops trying to fight it off. And those cells, they undergo a change. And the smoker no longer has the cilia, no longer has as many of those protective structures in place. So now more of those particles are going down deeper and deeper into the lungs. And 25, 30 years later, we call it cancer. Right? So basically the body gives up. It can no longer fight off those changes. So as a smoker's epithelium changes, we call it a metaplasia. It can revert back. Okay? After someone stops smoking, there can be some reversion. Hypertrophy. Hypertrophy. We're talking about an increase in size. An increase in size of an existing cells to undergo hypertrophy or hyperplasia. Plasia, you now see hyper means an increase in the number of cells due to mitosis. So more cells are dividing. So hyperplasia, an increase in the number of cells due to cell division. Neoplasia, we mentioned a neoplasm. A neoplasm is a new growth, usually a tumor growth, something that's unexpected or is out of control. So new growth, neoplasia. And then atrophy is a term that refers to both the reduction in size and number. Anyone here ever had a cast on their arm or leg for 6 or 8 weeks? And you took the cast off, and tell me what you experienced. A lot smaller, right? Yeah, so your cells, your muscle has undergone what? Atrophy. There is literally a decrease in the number and in the size, and after a while you start exercising that muscle and that limb back, and the muscle starts to match the other side. I wouldn't want to live in a cast for 6 weeks, but I'd love to see what would happen. Right? Put me in a body cast for 6 weeks, and I'd come out a little skinnier than I am. I think that'd be a great thing. Now, what about necrosis? Necrotic tissue is dead, dying tissue. But this is not dead or dying tissue because it's normal apoptosis. Or what's apoptosis? Programmed cell death, right? Cell death that's supposed to happen to maintain your overall body shape and metabolism. But necrosis would be tissue death from some sort of damage. So a burn, a scalding, some sort of damage, and the cell death is not always reversible. So it's something that you're going to lose. A tissue becomes necrotic, and you might lose that tissue. It cannot be replaced. Okay, so that brings us to the end of that chapter. All about histology, recognizing your 16 different images, knowing what those tissues are, why they're there, and a few special terms about tissue changes. What, if anything, in there can I restate for you or clarify? That's chapter 4 in your new book. And you know what? We're finally, you might be saying, wow, we are finally to anatomy. It took us, remember this should be chapter 5. We'll change that number. This is chapter 5 in the new book. But you signed up for anatomy class. You finally are labeling some bones in lab. And we're finally talking about our first system, the integumentary system. Everything up till now has been getting us up to this point of understanding systems. We talked about atoms and molecules and macromolecules and cells and organelles and tissues. And finally, we're at our first organ and organ system. So let's move into this. And this will be our last little bit of information for the exam. here's a classic picture of the skin. You'll need to be able to recognize and label this for the things that I tell you right now. So I'm looking at this, and I see the outer layer of the skin, epidermis. Right, so there's the epidermis. We know that it's all of this layer from here up. And how would we describe the epidermis? It's made up of what? What kind of tissue? Epithelial, more specifically. Stratified, squamous, keratinized. Keratinized because the skin it's dead, right? It's at the surface, it's dead. It's filled up with this protein called keratin. So we see multiple layers all the way to the top. What's everything below that? So that means that my line is on the basement membrane, isn't it? So my little pointer right now is moving on the basement membrane. Deep to the basement membrane is what? A layer called the dermis. But we could also say that the dermis is made up of what? Connective tissue, right? It really is a connective tissue layer, a pretty thick connective tissue layer. And in this connective tissue layer, there's some muscles that we'll deal with later. There's some oil glands, there's some sweat glands, there's all sorts of things embedded along with some blood vessels, some nerve endings. And all that is down here in the dermis. Now below the dermis, that's all there is to skin. Skin is just those two layers. Skin is only made up of your epidermis and your dermis. Under the skin is the hypodermis. Right? So it's not officially a part of your skin. It is instead a layer, mostly of fat, that your skin sits on top of. So it's referred to as the hypodermis or the subcutaneous. A moment ago I called it the cutaneous membrane, right? What was the cutaneous membrane? Skin. It was the epidermis sitting on the dermis. So this is the layer underneath the cutaneous membrane, underneath the skin. And we see down here a lot of yellow representing adipose. There are other nerve endings in here that we'll deal with more specifically when we get to the nervous system. But for now, know the layers of the skin, and I'll add to your understanding here in the next couple moments. Now, the skin can also be referred to as your integument. That is everything that covers your body. But it's not just that. So you've got your skin, you've got what's called the cutaneous membrane, but the integumentary system as a system includes not only your skin, but also those structures that are derived directly from the skin. So that's going to include, for example, your fingernails, your hair, your sweat glands, and your sebaceous glands. Now, sebaceous glands are glands that release sebum, and that is oil. So these are your oily glands, right? So we have sweat glands that are more watery, and then we have sebaceous glands which are more oily in their secretions. So let's talk about the integument. It is your body's largest organ. It varies from about 1.5 millimeters in thickness to 4 or more millimeters, especially if you've got calloused palms or calloused fingers, calloused soles of your feet. It is protective. We recognize that this is a stratified epithelium, and those stratified epithelia, whenever you hear stratified, think protective. And we know that right below that epithelial layer is the underlying connective tissue, and down here we find lots of blood vessels. Are the epithelial tissues of your epidermis alive? Some are. The lower levels are alive. The upper levels die and become flaky, right, and become the dead cells. But the lower cells are very much alive. But remember, epithelial tissues do not typically have blood supply. So where must they get their blood vessels or their blood supply? The answer is down here, right? They're going to get it down. The blood flow is going to be coming through the dermis, and there's going to be diffusion of those nutrients and the oxygen to the epithelial cells. So the connective tissues are providing the blood flow, the nutrients, the oxygen, right? The ability to get rid of waste products, all that's happening. Plus the connective tissue has a lot of collagen in it, which gives our tissue strength. And the connective tissues have elastin in them, which gives our tissue some elasticity. So we've got all those features that we've talked about in connective tissues. There's also, though, going to be in your skin some muscle. So not only do we have epithelium sitting on top of, or epithelia sitting on top of connective tissue, but there's also some smooth muscle in your skin. These muscles are going to help control the diameter of your blood vessels. And there are also these cute little muscles that attach to each hair, these erector pili muscles that I'll show you in a moment. And so we've got muscle within our skin. And finally, there's lots of nerve endings. Right? You can tell hot from cold, from painful to light touch to higher pressure. So what am I trying to convince you of? The integument is a what? It's an organ, right? And what is it? What are all organs composed of? Tissues. So that within the skin, what do I have? Epithelial tissue, connective tissue, muscle tissue, and nervous tissue. So no matter what organ I hand you, I can hand you a brain, a heart, a kidney, a liver, or a chunk of skin. Somewhere within that organ, I'm going to find some epithelial, some connective, some muscle, and some nervous tissue, right? Because we know that tissues combine to make organs. So again, that hierarchy of atom to atom, we can't forget about that. We know that there are two layers. This is not news for us. We know that the outer layer of the skin is stratified squamous, keratinized, even more specifically, and we would call this the epidermis. Down below, in the dermis, is dense irregular. Now, I did not have you recognize dense irregular. Right? We did learn what dense regular looks like under the microscope. We did not, and nor do you have to right now, know what dense irregular connective tissue looks like under the microscope. But I want you to know in your head that the dermis is largely dense irregular. There's also a little bit of areolar down there and a little bit of fat down there. Okay? So together, we've got all these different layers in our skin. Let's take a look. I think you're going to find this to be very straightforward. Let me draw for you the basement membrane. So what does that mean? Everything above that line is epithelial. Everything below that line is connective. In this particular picture, the connective tissues look like wood grain, don't they? Kind of looks like kind of a woody coloring. And what I have now are multiple layers of epithelial cells going up to the flaky, dry layers at the surface of the skin. Now there's layers here. I'm going to introduce these layers to you one at a time in a few minutes. But you'll see them over here. They're all called stratum. One layer stratum, many layer strata. And there are 5 layers here in the skin, specifically in the epidermis. There's a stratum basale, spinosum, granulosum, lucidum, and corneum. I'm going to have a slide for each of these and describe what each layer is unique for. Oftentimes, the cells here in the epidermis are called keratinocytes. Keratinocytes. Cells that are going to make what? Keratin, right? Remember, this is keratinized stratified squamous. So these are cells that are going to or have made this protein called keratin. So you'll see them called keratinocytes. It shows us here that the cells deeper are living, right? They're living keratinocytes. They're carrying out their metabolism. This slide even shows us a cell here that's undergoing division. Do you see the chromosomes? There's another one. Another anaphase going on here. So these cells are actively dividing. And then what happens is that the cells begin to undergo apoptosis. As they move toward the surface, they become dry, flaky cells. So what else does the skin do for us? And I think half of these we know, and then we'll come back and look at the layers. So we recognize that the skin is a protective layer. It protects us against chemicals and trauma, temperature extremes. It prevents us from losing too much water. We still lose, though, about 500, different books, 250 to 500 milliliters of water every day just through our skin through a process very similar to evaporation. We're regulating our temperature of our body. If you go to a nursing home, what's the number one complaint of the residents? And maybe of the people working there? It's too cold, it's too hot, it's too cold, it's too hot. What's the problem? As one ages, their blood vessels in their skin are no longer as able to adapt and to help regulate body temperature. So there's a lot of loss of body heat and inability to regulate body temperature because of that change. There's also vitamin D made in your skin, or at least it begins there. And without your skin and without the proper molecules there and without UV radiation, we wouldn't have vitamin D. Most of us in the wintertime are vitamin D deficient because we don't get enough sunlight to our skin in Michigan. We have in our skin some of our immune defenses. There are special cells, and I'll show you where they're found in the layers, that are fighting off against invading bacteria or viruses. These cells were described by a guy named Langerhans. So these are called the Langerhans cells. This is also in the skin where we can feel hot and cold and pressure and vibration. So we have lots of receptors, nerve endings. And finally, we excrete, right? We sweat out salts and water and even some oil. So we're actively secreting molecules from the skin. So those are all the basic functions of the skin. Let me go through these layers one at a time from you. This is from the bottom to the top. We're in the epidermis. So right now I'm dealing only with the epidermis. The deepest layer is called the stratum basale. Looks like basement, right? The basal layer. Where else have I used the word basal before? Remember I said every epithelial cell has a apical and a basal surface. That is, it knows its top from its bottom. So the basal surface is also the bottom. So the basal, right, the stratum basale, the bottom layer of the cells. In this diagram, it's this one layer of cells. You can see them, it's just that one layer of cells. And they are mostly cuboidal, although there are some in there that are more columnar, not that critical, but it's a single layer of cells. This is the layer that actually separates the epidermis from the dermis. These cells are sitting directly on the what? These stratum basale are sitting right on the basement membrane, right? They're sitting right on that layer. And there are three kinds of cells in this layer. The most abundant are the keratinocytes, the normal cells, right? By far the most abundant. These cells will eventually produce keratin as they move toward the surface. Number two, though, there are melanocytes here. Cells that produce what? Melanin. Melanin is the protein, the pigment, that gives you your skin color. You've heard it said that color is only skin deep. Well, it's not even skin deep. Your skin color is only epidermis deep, right? It's really only at the epidermal layer that your skin color is established. So these cells produce pigments called melanin. We'll talk more about it in a moment. They're found in the stratum basale. Lastly, there are tactile cells. They're also called Merkel cells. These are sensory nerve endings. Remember, what's our universal color for the nervous system? Yellow. See some yellow down here? It's not very distinct, but see yellow coming up right there in that blue cell? So there's yellow coming up. That is a nerve ending. It's coming up. It's in the stratum basale. And this kind of nerve ending is going to allow you to feel very sensitive changes. So it makes sense, right? The nerve ending is very close to the surface. So this is the kind of nerve ending that would help you distinguish soft from softer, or silky or from really silky versus not silky things. So very fine differences in touch. So that's the stratum basale. Let's move up. Stratum spinosum. Stratum spinosum. Now this is the more pink layer in here. The stratum spinosum is all of this layer. These cells are very much alive and well. They are dividing. They are metabolically active. They kind of start changing their shape. So the shape of them I'm not too concerned about. But also visiting in this layer are these cells called the Langerhans cells. You see it here in yellow. And that Langerhans cell is a macrophage. Macrophages are cells that can attack other molecules. So it's a macrophage. And macrophages, again, this dendritic Langerhans cell is a macrophage. And these cells can gobble up viruses or bacteria that were to try to get into or past your epidermis. So that's your stratum spinosum. The next layer is the stratum granulosum. I just use that word gran in vocab. A granular appearance or a particulate. And what you're going to see here is this dark layer. And this is a layer when the cell is literally, the cells are beginning to die. The cells are actively undergoing apoptosis. And as the cells die, their organelles are being destroyed, their DNA is being destroyed, and they become granular in their appearance. So this one's kind of easy to remember. They start looking odd. And what they're doing, the last big thing they do, is these cells produce lots of keratin. Remember, keratin is that waterproofing protein that's going to provide protection to our skin. And the cells start becoming thinner, and again, they look more granular in their appearance. Stratum granulosum. Pretty obvious when you look at a model or any tissue. You'll see that it's only 3 to 5 layers thick, but you'll see a color change happen between the living cells and the dead flaky cells. And that's the stratum granulosum. The stratum, I missed one. The stratum lucidum. What does lucid mean to you? Lucid. If grandma's lucid, her mind is still clear, right? If you've heard that expression, I don't know, right? So someone who's still lucid, they're still clear, their mind is still clear. So that's what I think of. The stratum lucidum is a lighter colored layer. But the stratum lucidum, we can say it's more translucent. It's very thin, only 2 to 3 layers thick, and it's only found in what's called thick skin. And I'll describe thick skin to you in a moment. But thick skin is what you find on your palms, on your soles of your feet. Okay, so the palms, I don't know what that means. Feet, not heat, right? So the soles of your feet. Okay, so that's your thick skin, and that's the only place you're going to find this stratum lucidum. There's a different protein here that gives them this translucent appearance. I'm not going to ask you to memorize this word, so don't worry about that. But just know that as the cells are making lots of keratin, they become more translucent. But this is only in your thick skin. Can you see it here? Very thin layer, more white in appearance. Then finally, there is the stratum corneum. If you have a corn, you have that outer layer of thick skin. And so the stratum corneum, all of the outer layers, these cells are dead. They're more superficial. They have filled up with keratin. As they get shoved to the outer edge, they get very flat. So in shape, these cells are very squamous, many, many layers, stratified, filled with keratin. And sometimes you'll see the word corneocyte to name these cells, cells that are part of the stratum corneum, corneocytes. One of the characteristics of these cells is that as they underwent apoptosis, they lost their nucleus, right? They're no longer living. They don't have a nucleus. So they're anucleated. So when you look at the surface, you'll see flat cells, but you won't see any evidence of a nucleus. Another good sign under the microscope that you're looking at stratified squamous keratinized. It takes about a month for the cells to go from the stratum basale and move their way up through the layers. So every month you've got a brand new layer, basically, of cells that you're constantly renewing, but it takes a whole month for all of the layers to move through. Those are the layers. Any questions there? It's not too bad. A little bit of memorization, but basale, spinosum, granulosum, lucidum, and then the corneum. Let's continue to talk about this a little bit, and we'll compare and contrast. I mentioned thick skin to you. There's thick skin and there's thin skin. The majority of your body is described as having thin skin. Right? Thick skin is only going to be found on your palms and on your soles, your fingertips, and we'll just keep it there for now. So we have the thick skin. And what you're looking at here in this image, that little blip down there, that's your connective tissue. Everything above it is epithelium. So can you now see the layers? I know it's labeled here, and I know it's kind of small, but the very first layer would be what? Stratum basale. And then multiple layers of living cells, stratum spinosum. And then that very granular layer, stratum granulosum. And then a very thin clear layer, do you see it? Stratum lucidum. And then the rest of it, look at it, hundreds and hundreds and hundreds of layers of corneum, right? The stratum corneum. So we're looking at a thick epidermis here. Now I'll compare this in a moment with thin skin. Now this can be rather thick, okay? And again, thick skin has all 5 strata, all 5 layers. But there's something different about your thick skin than the rest of the skin in your body. What's missing on your palms and on your soles? Hopefully you have no hair there, right? Right? You're an exhibit in the zoo if you do, right? So you don't have any hair on your palms. So that's a difference. Our thick skin doesn't have hair. But what else is true about your palms and your soles? They sweat a lot, don't they? But do they sweat the same as the rest of your body? Thankfully, our palms and soles have normal sweat glands, watery sweat glands, but we don't have any oily, at least not a lot of oily sweat glands on our palms, do we? We certainly don't sweat from our palms the way we do from our underarms, and for that we're thankful. Right? Can you imagine that world? Right? Okay, we would do a lot more Eskimo kisses than shaking hands, right? If we had oily hands. Now let's compare this side by side. On the left is the same image you just saw. This is thick skin. Over here is, I guess 2 letters don't help. Thick skin. And over here we have thin. Now what's the difference here? Let me get your bearings on this. The basement membrane is here. Okay? So all of this down here is what? Connective tissue. That's the dermis. You now appreciate side by side how much thinner this thin skin is. There's still a stratum basale, 1 layer thick. There's still a stratum spinosum, multiple layers thick. There's still going to be a layer where the cells are dying, so there's still going to be a stratum granulosum. There will not be a stratum lucidum. And the stratum corneum is very unimpressive. It's not very thick at all. The majority of your skin on your body looks like this under the microscope. What else do we see? All over your body, even where you can't visibly see it, there's going to be hair. So everywhere on your body, even if it's microscopic, there's hair. There's only a few places on your body where there isn't hair. Besides your palms and your soles, that would include where else? What other special places have no hair? Well, thankfully on the end, but out, right, your lips, right? Nothing on the lips themselves. And the external genitalia, those are really the only other places where you are truly hairless. So just know that the 2 layers here, the biggest difference is this thickness of the stratum corneum and the fact that thin skin does not have a stratum lucidum, right? No stratum lucidum in thin skin. Again, though, lots of hair follicles. Sebaceous glands, those are those oily kind of secretions. wherever you have hair, therefore, you have oil, right? Right? Wherever you have hair in your body, you have oil secretions, and that's a good thing. You want your skin to be supple. You want your skin to be moist. You want your skin to not be too dry. And along your hair follicles, we'll see, are where some of these oil glands are. So you want, where do we get our skin color from? I've got different examples from around the world on here of individuals with different skin colors. What do we have here? Bottom right looks rather what? Where do you think this young lady's from? Muskegon. Red hair, rather pale, right? Maybe a little Irish or a little, Northern Isles or something in there. And then we move over and we see much darker complexions. And again, this whole rainbow of colors. In the bottom left, I've got a picture of siblings, right? So I've got an African child, and on his back is his little sister, and she's albino. So what's the difference there? Her skin, through a genetic mutation, does not make melanin. So her melanocytes produce the same pigment mine do, like nothing, right? I don't produce much melanin. I'm very, very fair. Other individuals produce different forms of or different types of melanin, giving us the colors of brown and black and red and tan and all the different variations we have on our planet. So what gives us our skin color? Actually, a combination of three things. The most is truly your genetics, right? Your melanocytes. That's by far the greatest indicator of your skin color. But I also want you to realize that when you're embarrassed, right, or when you're exercising, there's more redness to your skin color because there's a protein in your blood called hemoglobin. We'll deal with it later, but hemoglobin is red and does provide a reddish color to our skin, especially, again, when we're embarrassed or when we're exercising. And if you eat enough vitamin A and carrots, you will actually get sort of an orangey tinge to some of your skin. So it is possible to get an orangey tinge from carotene. Now look at the spelling of that protein. That is not keratin, the stuff in our skin. It's carotene. Unfortunately, sounds the same, looks very different. And again, we have a lot of this in carrots, vitamin A. So carotene. Now, why do we need melanin? Why do we need it? We don't need it, but what are the advantages of having melanin? If you have a darker complexion, what do you not do as easily? Burn. So what does melanin do? It helps to protect us against UV radiation. Think about the world for just a second. Where do people with darker complexions come from historically? From the equator, right? From the equatorial areas. Where are the pale folks in the world? At the poles. Okay? So, I'm telling you that melanin is protective against UV radiation. It can protect against skin cancer. So, where in the U.S. do we have greater skin cancer problems? Not up here, right? Greater skin cancer issues in the desert Southwest, where people are outside and they get a lot more UV radiation. Where in the world is the highest skin cancer rate? Where do you suspect in the world has the highest skin cancer rate? Australia. Why? A bunch of displaced Europeans living in the equatorial areas. Right? So in northern Australia, they're closer to the tropics. But historically, these are Europeans, right? They're English folks who came over, and so their bodies have not adapted to that ultraviolet radiation. And so the highest rates of skin cancer are especially in northern Australia. And that makes sense, right? Because it's a bunch of white people living in tropical areas. There are other politically correct ways to say that, I know, but I'm not one of those PC folks, right? So just a bunch of displaced Europeans, right? I could have said criminals, but that's another history lesson. Now, what else do we see in our epithelial layers? I told you that typically, or maybe on your own body, what are some of the different markings that you might see on your own body or those of your family members? So some people have moles, and another name for a mole is a nevus, and this is a benign overgrowth of cells that are melanin producing. So you tell me, what kind of cells are these that make a mole? Melanocytes, right? Those melanin-producing cells from the stratum basale, they begin to grow. Sometimes it can be a problem, right? If you go to the dermatologist and you already know you are prone to having moles, they should be checking you out, right? The dermatologist should be checking over those moles. Occasionally, those moles can begin to grow a little bit more out of control, and the dermatologist or your general doctor should be cutting those off, sending them off to biopsy, making sure that there's no metastatic changes to those moles. So they're kind of a pre-cancerous thing that we need to be watching. Doesn't mean you're going to get cancer, but it is a benign overgrowth. And definitely moles can become more malignant with exposure to UV. So if you already have moles, you definitely want to avoid ultraviolet exposure. Freckles, areas of high melanocyte activity. Not the number of melanocytes. I don't think I said that back here, but it doesn't matter where you are in the world, your number of melanocytes is the same in all individuals. So I have just as many melanocytes as individuals from Africa. The difference is what? Not the number of these melanocytes, but the amount of pigment, the amount of melanin that's being produced. So my cells essentially don't have melanin, and other people have various types of. Melanin comes in two basic types that give us the yellows and reds and tans and browns of the world. Okay. So again, we have moles, we can have freckles, and then there are hemangiomas. Now break that word down for me, folks. We know angio, blood vessels, right? And oma is, we'll learn later, is more like a tumor or a growth of some sort. And then what is hemo or hemato? Blood. So what is this? These are blood-filled vessels really close to the surface, and sometimes they're more temporary, sometimes they're more permanent. They come in two basic types. There are capillary hemangiomas. These are oftentimes present at birth, and they're called strawberry birthmarks. I think 4 of my 5 kids had these strawberry birthmarks. They were little clusters, kind of looks like a little strawberry cluster near the surface. One daughter had it on her back, one had it on the back of her head. My little guy had one on his chest. It really stuck out quite a ways. And every one of them, by the time they started kindergarten, preschool, they were gone. So this is more of a temporary thing. During development, these blood vessels are toward the surface. They tend to rescend down and usually disappear early in childhood. But there are also cavernous hemangiomas. These are the port wine stains. These are typically permanent, much more extensive, and for obvious reasons are much more devastating as far as a birthmark goes when they're on the face. So this is Gorbachev, right? His little mark on his head. This is Drew Brees, the quarterback. He's got one on one side of his cheek. Whenever he's doing commercials, he's usually showing his other side. So if you see him straight on, you'll see a little cavernous hemangioma on one face, on one side of his face. This up here is a pretty extreme case of a strawberry mark, but likely that would all be gone by later, by kindergarten. So those are just some different markings that you would see on the surface of your body. Now, we also have fingerprints. We know this. This is nothing new to you. In an anatomy book, those are called friction ridges. Right? Why do we have fingerprints? Well, to help the police catch the bad guy, but also to help us grasp things. So the dermis and the epidermis, as you're going to see in the pictures, tends to look like peaks and valleys. And the epidermis and the dermis don't sit on top of each other like a flat layer. So there's a lot of peak and valley going on that helps to give us a gripping that we call fingerprints. Now, there are all sorts of these fingerprints. I'm not going to ask you to know these, but if you watch any kind of CSI show, there's arches and there's whorls and all sorts of different structures that are on our fingerprints. It's completely random. Even identical twins do not have the same friction ridges. Can you change them? Only by what? Burning or damage or chemical damage, right? So you basically strip away the top layers of the skin, yeah, you might scar up or change those fingerprints, but that's a pretty significant amount of damage to have that happen. So again, we're not going to worry about those particular patterns. So that's everything to do with the epidermis. Let's go down to the dermis now. So we know this is a layer lying deep down from the epidermis. It is composed largely of connective tissue. We've got elastic and reticular fibers down here. We've got collagen fibers giving lots of strength. This is also where the blood vessels and the hair follicles and the nerve endings and all the other business of the skin is going to be found. 2 layers of your dermis. 2 layers, shown here nicely. So let's make sure we see the epidermis first. So here we see those ridges. See how the epidermis is very much peaks and valleys, right? Up and down, up and down. Looks more like an egg carton or a mattress pad. And then as you pull away, the top layer of the dermis is called the papillary layer. The papillary layer. That's the more superficial layer. And the deeper layer is the reticular layer. The reticular layer. So we see that word reticular in a lot of places. It usually means something's building on it, right? A reticulum, a layer of some sort. So we have the papillary and the reticular layer of the dermis. Now, the word papilla, we're going to see this term also in many, many places within the course. But papilla means nipple-shaped. And we'll see this term over and over in different places. So you'll see that, right, these layers have these upswinging peaks and valleys, sort of nipple-shaped structures. And that's where it gets its name, the papillary layer of the dermis. This is largely loose areolar connective tissue. I know I told you that the dermis is largely irregular, but at this little thin layer called the papillary layer, it is mostly areolar or loose areolar connective tissue. This is the layer that interlocks, right? Interlocks, interdigitates with your epidermis above. This is also where some of your touch receptors are, your nerve endings that allow you to feel fine touch. So, for example, you can see some yellow structures up here in the papillary layer, and those are some of the nerve endings that allow you to feel really fine touch type things. The lower level is the reticular layer. It's the deeper layer. It goes all the way from the papillary down to the hypodermis. And this is where most of that dense, irregular is. Right? And this is, again, where most of the business end of the tissue is. This is where the blood vessels and the most of the other nerve endings, the hair follicles, the sweat glands, all of that is down in the reticular layer. Here's a lovely thought. Let's talk about stretch marks. Wrinkles. What are stretch marks? Anybody have any? Nobody wants to admit it, right? What are stretch marks? How do we get stretch marks? At some point, your skin has been, quote, stretched, and it either kind of went beyond its limits, or it stretched, and when it recoiled back, it didn't go back quite the same way. What structure in your body gives your skin lots of strength? Whenever I say strength in tissues, you're thinking what protein? Collagen. And then you know there's elastin tissues also, or elastic structures that also let your body stretch. It can stretch to a certain point, but if your skin stretches beyond its collagen limits, it's basically like you've torn those collagen fibers, like tearing a tendon or a ligament, and it will leave a mark. So basically, you've excessively stretched the skin beyond its elastic limits. You've torn the collagen fibers. That's what's going to cause a stretch mark. Wrinkles, a little different. Wrinkles are going to be from aging, but specifically from exposure to excessive UV. So I always remind my daughters of this, that no tanning bed is completely safe. And that more UV radiation, you might look good now, but you're going to look a little bit more like a Shar-Pei later in life. Okay, so it's really important that you minimize your UV exposure. And if you want to see people who have had the worst UV exposure, I can remember going to a farm in in Arizona, outside of Tucson, to a cactus farm. And this woman lived outside all the time, living in the garden. And I've never seen such deep grooves. I mean, her face, such deep wrinkles from that excessive, overexposure of UV. So with that excessive UV radiation comes a loss of flexibility and an increase in the thickness of the dermis, which leads to those deeper wrinkles. In your skin, you have what's called lines of cleavage. Now, if you look at this image, right, it just looks like a bunch of lines, but a surgeon, especially a plastic surgeon, but any surgeon is going to be aware of these lines of cleavage. And basically what they are is this is the angle of the collagen fibers. So as you look at different layers of the skin, you can see here across the abdominal region, the lines are more perpendicular. And along the legs, they're a little bit more sort of like a winding, like a barber pole. And if you were going to have surgery, and it were elective surgery, right, and you were choosing to have it, and you wanted to minimize the scar, the doctor would want to make sure that they were cutting the skin in the same line, in the same angle as these lines of cleavage. Because what will happen is that it makes it easier for the skin to heal up. So when you put the sutures in, when you put the staples in, it's not going to tend to pull apart. It's going to have a minimal scar. If, for whatever reason, either through trauma or there just is no choice, you have to have an incision that goes perpendicular to the lines of cleavage, then as this picture shows on the top, you're going to have the scar, the scar is going to be up and down, but the lines of cleavage are going perpendicular to that. And these are going to be the ones that are less likely to heal well. The staples will be pulling, the sutures will be pulling, and will likely leave a larger scar of sorts. Okay, so you want to be keeping in mind the lines of cleavage. You've also experienced this a little bit when you're cutting a roast, right? Sometimes you cut a roast in one angle and it just falls apart in other ways because, again, those collagen fibers are in a particular arrangement in muscle and in skin. Let me finish up with the hypodermis. We'll take a quick break. So innervation in the dermis, rather extensive, lots and lots of nerve endings here in the dermis. You're not surprised by that. We'll name some of those nerve endings when we get to the nervous system later in the course, but you have a lot of different nerve endings here. You also have all your blood vessels of your skin here. So when you're warm, what happens to your blood vessels in your skin? They dilate. We say the vasodilation occurs. So what happens to heat? Your blood vessels open up. You become more red in complexion. You're losing body heat, right? You're trying to get rid of body temperature. Whereas when it's cold outside, your vessels vasoconstrict. They become smaller in diameter, less blood flow to the surface of your skin. You become more pale, right? You're conserving heat inside your body. So our skin, because of this vasoconstriction and vasodilation, is huge in regulating our overall body temperature and our comfort level. So that's what I just said here in the vasoconstriction and vasodilation story. Again, this ability to control this vasoconstriction and vasodilation decreases with age. So the elderly are oftentimes complaining about the lack of temperature control. And then let's go down to the hypodermis. We know it as the subcutaneous or the hypodermal layer. It's deep to the dermis. It is not officially part of the integument. It's below the skin. It is largely that loose areolar as well as adipose. Now, where you have this adipose or where you have this subcutaneous fat is somewhat gender-related, sex-related, and partly genetic. So if you look in your family pictures, you will notice that, or go to the mall and watch a family walk down through the mall, and you will notice that some family members have the same body composition, right? So you might see people with, well, I grew up with a disease. My mother always said I had Fatima-thigh. Now, I didn't know what Fatima-thigh was, but I knew I had it, and everyone in my family had it. And she always described that in the Macauley household, we all have Fatima-thigh. Well, I finally figured around age 10 or 11 that meant fat on my thigh. And if you look on all of the family pictures, sure enough, we all have phatomythi. So you go to the mall and you watch, right? And you'll see families, and they all share some of those special characteristics. Part of that's genetics, right? You just were born with that triple chin. Thank you, Grandma. And that chin will be with you no matter what you do, right? You'll have that little extra flap there, and you just say, thank you, Grandma. And that's a genetic thing. And then, of course, women are going to accumulate typically more fat in their breasts, in their buttocks, in their hips, and guys in different places, okay? So it's going to be different. Again, there's a large genetic predisposition to where it will accumulate. You have some control over how much will accumulate, but you don't have a whole lot of control over where it will accumulate, and that's what keeps plastic surgeons in business. Let me stop there for about 10 minutes, give you a little break. We'll come back and just finish up the last little bit on skin, maybe about 7 minutes, not too long. Okay, so let's finish up here this last little bit on chapter on the skin. And there's a couple of things here I'm not going to get really terribly excited about, and one of those is nails. I just want you to know that your fingernails are a scale-like modification of your epidermis. So what does that tell us? The epidermis is a bunch of cells that contain what? Keratin. And so, too, your fingernails are made up largely of the protein keratin. So just recognize that it's pretty much the same stuff. Keratin is making up your nails. Keratin is also making up your hair. So your hair and your nails are also made up of the same stuff. It's really just an extension of your stratum corneum, right? Remember the stratum corneum, the outer layer of your epidermis, which is pretty much all layers of keratin. As far as structures of the fingernails, you labeled a couple things in lab. I don't have a model of fingernails, and I'm not going to get excited about it at all. So if you're really interested in your fingernails, you can take a greater look at this, but nothing I'm going to quiz you directly on. Just know the keratin. When it comes to hair, again, we know that hair is found all over your body except in places where you have thick skin, plus your lips and external genitalia. And a single hair is called a pilus. And we'll see the term pili, pilus, other words like that. That's just a single hair-like structure. Now you have different types of hair on your body, and there are different functions for hair. Number one, protection. We get that. It protects us from UV radiation. It protects our ear and our nose. We got eyelashes and eyebrows, all those kinds of things that protect us. Certainly we have heat retention going on. First thing we do with a newborn is put a little hat on them. They don't have as much hair usually. That helps to maintain some heat. We see each other's facial expressions largely through their eyebrows and eyelashes. We've also got touch receptors in our skin and our hair also. If you just run your finger over the hair of your body, you're very much aware that you're touching your hair, so your body. So as you touch little hairs, you're also detecting that through your nerve endings. And finally, we do, as humans, release pheromones. It was not always known if humans had pheromones. We knew that animals had pheromones, but clearly now we know that humans also have pheromones, and these pheromones are helping to, there's signals that we send out from our sweat glands that help to influence those around us. There are 3 basic kinds of hair on your body, or at least during your lifetime. Lanugo is a very fine, unpigmented, downy-like hair. If you've ever seen a preemie, right, a premature baby would be born with some of this lanugo on them. For the most part, most of us have lost this by normal term. So normally a full-term baby, you wouldn't see this. Most of the hair on our body is just called vellus, regular old body hair. And then there's that coarser hair, that terminal hair. Eyelashes, eyebrows, axillary hair, pubic hair, facial hair after puberty. So those would all be considered terminal hair. Now, again, I'm not going to get terribly excited about hair structure, but I do want to take a look down in the dermis and right down in this area. And there's a bulb down there, and that's where these cells originate. And there's a papilla. Again, there's that word papilla, a little nipple-shaped structure. And within that little nipple-shaped structure, there are nerve endings and blood vessels. And there's a little root down here. Here is that erector pili muscle, cute little muscle that's attached to each of your hairs. So when you're scared, that little muscle contracts and all of your hairs stand upright. We also see here this sebaceous gland, this big gland structure. And this is secreting some of that oily kind of secretions. So you'll see these sebaceous glands are right up along your hair shafts. And we want that. I mean, we want our hair to have some suppleness to it so that when you pinch your skin, it doesn't stay there, right? It lays back down very, very quickly. Now, I'm zooming in to that papilla region. And there's that little papilla region way down here in the hair bulb. And here's an electron micrograph of a hair coming out of all these flaky layers, right? So you don't realize how dry looking your skin is till you look at it under electron microscope. And here you see those dry layers of the stratum corneum with a hair follicle sticking up through the middle. Now, where does our hair color come from? We mentioned our skin color, but also down deep in the papillae, there are melanocytes. Okay, so again, we talked about melanocyte cells that make melanin, and you also have melanocytes deep in your hair follicle. And gray hair is a gradual reduction of those melanin-producing cells. So the hair becomes grayer, and then white hair is a complete lack of that pigment. And that changes with time. It can also be brought on by hormonal changes, by environmental changes. Pregnancy can change hair color a little bit. Other things can also change color of hair. There can be dietary issues, fever, stress. All of those things can cause one to lose hair. Hair is typically going to grow somewhere around 1/3 of a millimeter every day, and it's going to continue to grow for 2 to 5 years. Really, really long hair actually stops growing as much. And you're going to lose, on average, somewhere between 110 and 100 hairs. Anything over 100 hairs a day, you may have what's called alopecia. Let me go back, sorry. And alopecia is a thinning of the hair, which is typically a result of aging. It's I guess, almost become chic now. But for guys, right, they start losing hair and they take it all off. But a little bit more concerning usually for women who have thinning hair. Sweat glands, lovely thought. It's tough to find great pictures like this. But the sweat glands of the body, there are a couple different types. Let's just think about good old sweat glands. There are the normal sweat glands. When I say normal, I mean these are your watery kind of sweat glands, your merocrine sweat glands. We talked about how they secrete. They stay merry, they're alive. These are very watery glands. Huge number of these glands on your hands. Remember we discussed that your hands and feet sweat more. These are regular watery secretions. It's 99% water. There are also apocrine sweat glands. These are where your hair follicles are greater in your armpit, around the breast, and the pubic regions. And so these sweat glands are producing a thicker secretion that bacteria love. And so the bacteria start eating away at these secretions, giving us a distinct body odor. And we know now that your body odor is much like a chemical fingerprint. It is distinguished one from another. So those pheromones, the idea that each of us has our own unique body odor, is very much true. So if I take a look at skin here, what I'm going to see, hair. So I know I'm not where? I'm not in thick skin. I'm definitely in thin skin. And here I see a sweat gland, and it goes all the way up to the surface. This would be a, quote, regular merocrine sweat gland. So this is the vast majority of your sweat glands. Then there are these sweat glands that are around your hair follicles. Those are the sebaceous glands. And those are the glands that are much more oily. And notice that they're right next to a hair. But then there's also this type. This is an apocrine. And the apocrine sweat glands are more found in your armpit. They're more found in your pubic regions and are the ones that really give you the stinky sweat. And you'll see that the apocrine sweat glands are also associated with hair follicles. right? So wherever you have a lot of hair, you're going to have more of these sweat glands, underarm, pubic regions, for those sweaty apocrine glands. Then most of your body also has sebaceous glands where you have hair. So again, sebaceous glands are producing this thing called sebum or oil, more common with face and all over the body. I mentioned to you already that these glands secrete via the method of holocrine. That should be an O, not a A, but holocrine secretion. And holocrine secretion is going to be more prone to fill up our sweat glands with the sebum and create some blockages that can lead to boils and other skin problems. Most of these sebaceous glands are rather inactive until puberty. So an 11-year-old boy doesn't stink as much after soccer practice as when they're 13, right? So they really start to reek more when they get to puberty, when they start getting those sebaceous glands working in high gear. It's also when the complexion goes from beautiful to a little bit rough during teenage years too. I'm not going to have you recognize these images, so I'm going to let you put a gentle X through this. These are those sweat glands, but I'm not going to have you recognize the histology of them. Now, there's a couple other glands in your skin. Ceruminous glands, found, thankfully, only in your ear canal. Right? These are the glands that make cerumen. Cerumen is a fancy word for your earwax. It's basically a modified sweat gland that produces a very unique product, and it's meant to line the inside of your ear canal and protect you against things coming in. There's also mammary glands. Mammary glands, the production of milk, really, you could look at mammary glands as being modified or specialized sweat glands. They produce milk under hormonal control. Any thoughts or questions, concerns about glands or anything in the skin so far? I've only got just a little bit left dealing with burns before we're done with this content.
So then our lips considered thick skin?
Our lips are not considered thick skin, but they are hairless. Okay, so the lips would not have a lucidum. Okay, they would not have a stratum lucidum. Anything else? Okay. Well, let's talk about what can go wrong here. So burns. Major cause of accidental death. It can be caused by heat, radiation, chemicals, electricity, you name it. All sorts of things can cause a burn. And burns can be a very significant injury and can lead to a loss of body fluid, followed by infection, and you also have to deal with all the scarred dead tissue. Now, burns come in three basic types. First and second degree burns are called partial thickness burns because they only go partially through all of the skin. And then there is third degree, which is considered a full thickness burn because it does go through both the epidermis and all of the dermis. So it affects completely both layers of the skin. Some books, some individuals will describe fourth degree burns. That is, the burn is so deep that you can see down to bone and muscle tissue. That's really just a really bad third degree burn. So I'm not going to describe fourth degree burns in a different way, but you will see fourth degree burns. I'm not going to teach that. Just know that it's a really bad third degree burn. Let's go through this. You know some of this, I'm sure, just from everyday experience. First degree burn, pretty much a sunburn. Okay, you're only affecting the epidermis. You're only damaging some of the outer layers. You're not going down into the dermis. Characterized by redness, a little bit of swelling. Certainly can be painful, right? But it's only affecting the epidermis. Then there is a second degree burn. It is going to affect all of the epidermis and go down and partially damage the dermis. And the number one thing here is that it's going to form blisters. So when you think of a blistering burn, you're thinking second degree burn. The sun can cause a second degree burn. You can definitely get a really bad sunburn and get second degree burns with blistering. This is also finger on the stove and curling iron to the forehead, right, ladies? So this is a second degree burn when it blisters up. Very painful. right? Very painful, because what have you affected? You've gone down and you've damaged the nerves. So lots of nerve endings in the dermis. You're going to know it. Scalding, another example. Now, the third-degree burn is oddly painless. And the reason is, at the initial insult, the epidermis and the dermis were completely destroyed, as were all the nerve endings. So as soon as those nerve endings are gone, there's no sensation of pain in that localized area. Now, around the edge, it'll be very, very painful, because there you've got second-degree burns around the edge of a third-degree burn. But in the very center of a third-degree burn, you would not have a tremendous amount of pain or sensation. For a third-degree burn, a lot of the tissue is gone. And if you've ever had a scab, you tell me, how does your skin regenerate? Your scab gets smaller and smaller and smaller, right? So it's regenerating from the edges. And if you look at the depth of a scab, you'll see it gets less and less deep. So it's generating from underneath, it's bubbling up, if you will, and it's coming from the edges. Well, what if you destroyed all of the dermis? You no longer have the ability of it to bubble up from the bottom, and you've damaged everything else. Then we might have to do a skin graft. And a skin graft is basically taking a cheese grater to the thigh, right, taking some skin and layering it over and hoping for it to graft, to take, and revitalize itself. So in the pictures on the far left, there's a simple sunburn. In the middle, you see the blistering. Think second degree with blistering. Third degree, not much to look at here, but I would assure you that this would be can be a very large part of the body. With a third degree burn, the biggest concern is dehydration, because you've lost the barrier of the skin. If a person has a large percentage of their body with a third degree burn, you can give them an IV, and that fluid is just raining out into the floor, onto the bed, because they've lost their outer layers, and so that fluid is being lost from their bodies. And now they've got the opportunity for infection. So the number one problem is volume and blood loss and water loss, and then after that you've got the problem of infection setting in because you've lost that outer barrier. So again, just a quick commercial. First, second, and third degree burn, most obvious in the layers of the skin. First degree, epidermis, partially destroyed. Second degree, all of epidermis and some of dermis. Third degree, all the way through, full thickness, down into the subcutaneous layer. Survival rate. Okay, so someone has a significant burn. Maybe they were caught in an auto accident, the car caught on fire. Or maybe they were in a house fire. Whatever the situation. There are three things I want you to think about as far as survival of a burn. Has to do with how old you are. If you're 60 or older, that's one tick against you as far as risk factors. Number two, how serious is the injury? If it's more than 40% of your body, that's another tick. That's another major risk factor. And then number three, did you, while getting the burn, suffer any inhalation damage? So maybe you're in a burning house, or maybe you're in a car accident and you were got some burns, but you were in a seatbelt and you couldn't get out, so you're getting smoke inhalation during that time. So here's the deal. If you have none of those three risk factors, you have a 0.3%, I'm missing the word percent there, but you have a 0.3% chance of death. You're going to be fine, right? Higher chance of dying from walking across the street, probably. If you have one of those major risk factors, you're over 60, more than 40%, or suffered smoke inhalation, any one of those is going to bring your chance of death up to 3%. You're probably going to be okay. If you have two of those risk factors, you're at 33% chance of death. But the worst is if you're older, large area, and suffered inhalation damage, there's a greater than 90% chance that you will die from those injuries. So that's a little bit about burns. Any questions on burns? In a lot of my graduate research, we used to do burn studies. So we used to burn pigs. Pigs have skin closest to humans that we have in the animal world. So we would burn pigs and then study how those wounds healed and how they recovered. So it's a pretty gruesome area, but, pork rinds, good stuff. Little burnt pig skin. Anyway, on we go. Another lovely thought, acne, right? So acne, comedos, not very comedic, but comedos. And this comes in two types, black and white. Black heads and white heads. So what's a blackhead is a plugged up sebaceous gland, and it's plugged up with sebum, but it's still open to the environment, and that openness gets oxidized by the environment and turns black. So an open comedo, the blackhead forms, and so you have this oxidation of the white. If it's a closed comedo, it's below the surface, it's more swollen, a little more painful. It stays white because it hasn't broken through the surface and hasn't gotten oxidized. Okay. Now, oftentimes we'll see them right up along a hair shaft, right along a hair shaft where those sebaceous glands are releasing their product. So you always see those sebaceous glands, those big, ugly sebaceous glands next to a hair, and it gets clogged on the way to the surface. Now, I was looking for a beautiful picture of blackheads, and when I was doing a Google search, I found these lovely blackheads in the picture. And then when I was looking for a picture of whiteheads, I found this family on vacation. Okay, so there's blackheads and whiteheads. Now, the last thing, every system that I have for you will include what happens to the system as we age. Okay, what do we have to look forward to? Okay. So what's going to happen to our integumentary system as we age? Number one, it'll take longer for our skin to repair. Makes sense. Those cells down in the stratum basale are not as young as they used to be. They're going to take a little longer to repair. Should we get a scratch or a cut, it's going to take longer to repair. Number two, we'll become more wrinkled. Our skin will become less resilient. The collagen will be less organized. The elastin will become less elastic. So our skin will change its properties. Now, if you want to see how you're doing, do the grandma's pinch test. All right, so if you go to grandma, be nice to grandma. But if you pinch the skin on the back of her hand and you let go, right, her skin may not let go. It may just kind of stay there, right? So because as we get older, our skin gets thicker, and there's less elasticity, and it tends to be more wrinkled. And thankfully, when I pull my skin up, it still falls down rather fast, although it is getting slower. Okay? So you young folks, right, it'll just kind of go right back down to normal. Now, what's keeping that hair nice and supple, or that skin nice and supple, are the oil glands, right? Those sebaceous glands producing that oil. We want that. We want our skin to stay nice and soft. But as we get older, we'll have more wrinkles, we'll have less collagen, and we'll also be unable or less able to fight off infection. So should we get something in our skin, a virus or a bacterium or something, our skin will have fewer of those Langerhans cells, and we'll have less of those sebaceous glands, fewer of those sebaceous glands, leading to drier skin, right? So the grandma pinch test, the hair will stay up higher, or the skin will stay up higher. You'll also start seeing some changes in hair pigmentation, going white, or maybe losing hair, or maybe even adding liver spots. Liver spots are oftentimes found on the back of the hand. I'm getting a couple of them as I approach 50. And so I've got a few of these little liver spots occurring. These are just changes in pigmentation, usually from sensitivity to the sun. There's also going to be sweat production diminishing, so you can't sweat as much. This is going to lead to some problems with thermoregulation and figuring out how hot and cold you are. You'll have fewer hair follicles or none at all. You'll start making less vitamin D, right? So you'll have less ability to make vitamin D. This could lead to problems with your bone health, and in some individuals lead to real problems like osteoporosis. And finally, there can be the development of skin cancers from that extensive UV radiation. So as I finish up here in the last couple minutes, let me just describe to you the most common type of cancer. We already know that cancer is an out-of-control growth. We know that all cancer is genetic, in that all cancer has changed at the gene level, the ability of the cell to regulate its cell cycle. So the greatest exposure, or the greatest risk for skin cancer is exposure to UV rays. The more extensive and the more time that goes by, the greater the chance that skin cancer will occur. And it's especially more common in individuals like myself who have had severe sunburns. So the greater those sunburns you've had in the past, the greater you need to be more concerned about future skin cancers and be more proactive. So I can sit outside and get a second-degree burn in an hour. It's very, very sensitive. So I'll get blistering within an hour if I'm not covered up. So let's take a look at these. And you tell me, where are these cells within the skin originating? So a basal cell carcinoma, stratum basale, right? So this is a common cancer you'll hear about. This is the most common type. If you're going to get a skin cancer, this is the one you want because it seldom metastasizes. It usually is in a place where we can find it. It's on the face or someplace where it's visible, and we can usually remove it without issue. So if you're going to get a skin cancer, this is the one you want, right? Life is going to be fine for the most part. What you don't want to hear about is a malignant melanoma. A malignant melanoma is very deadly. These are the skin cancers you hear people dying of. They are arising from melanocytes in moles. So again, if you have moles, you want to make sure you're being checked out because those melanocytes are the ones that go nuts and can create malignant melanoma. They metastasize quickly. Now, the problem is they're at a higher risk for people like myself with histories of sunburn. So if you have moles and you have a history of sunburn, you don't want to play around with this. Every time you go to the doctor, make sure that they're examining you full body. Take it all off, spin around, have them document any and all moles that you have. Have them look at the diameter, the shape, the size. Compare it with the last time you were there. These things can happen so slowly that you don't notice them, but from a year's time, you'll see changes. Okay? And then finally, squamous cell carcinoma. Well, you tell me. These are the flat outermost layers of the skin, and they happen to be where there's skin exposure, back of the hand, scalp, ears, and they can metastasize. They're kind of intermediate. This is the one you don't want, right? Bad, bad, bad. And this guy will take it. Okay? You get to choose. Those are the ones you want. Okay? Now, the last thing is, come on, here we go. What are you looking for? So as you look at moles or spots on your body, be aware of the ABCDE, right? The things you're looking for in skin cancer. So number one, you're looking for asymmetry. If you see a spot, a mole, any kind of birthmark kind of thing, and you see it changing its symmetry, then you want to be concerned. If a normal freckle or mole is looking a little bit different, you want to know about it. Number two, is the border becoming jagged or irregular? Number three, the color. They normally are one color, and if you start seeing blotchiness, different colors popping up, you want to be careful about that or be alerted to that. Number four, the diameter. If it's enlarging, right? If it's getting bigger than a small pencil eraser, then you want to be concerned. And finally, elevation, if it's rising higher. Okay, so color changes, border changes, more jagged, rising, getting bigger. Those are all things that you don't want to mess around with. And you definitely want to alert your doctor to them and have them do a biopsy, cut it off, take a look at it, and make sure you're not at a higher risk for something more significant. Okay, so that brings us to the end of the chapter on skin. Our first system, exam on Wednesday. Full-length exam. Make sure you have an 884-E Scantron form. Cells. I mean, we talked about cells even before the first exam, didn't we? It's been a while. We had 1, 2, 3, about 4 full lectures on this material. So definitely look back through it. When is the mastering quiz due? Tomorrow at midnight. So that means you want to make sure you've done your mastering assignments and you've done your quiz. Now that quiz does count for a grade. So I would definitely do my mastering assignments first, make that practice, then do your quiz. It's not timed. It is open book. You should do pretty well on it. And then once you've done really well with that, then you'll come in and just do really well on the exam on Wednesday. Those who are in hybrid, be looking for an email from me that you have the option of taking it Wednesday or Thursday. But the rest of you who are in this class, I will see you Wednesday at 4 o'clock for the exam.